I'm Andy Ryland. I'm a crop protection consultant based in the Sydney Basin. This guide video has been made as part of the Building Biosecurity Capacity and Resilience within the New South Wales Vegetable Industry project, funded by the Australian and New South Wales Government Storm and Flood Industry Recovery Program, with support from AUSVEG. I'm going to talk to you about identifying pests and beneficials in vegetable crops and how I tie that into integrated pest and disease management programs. The critical part of any pest control operation is monitoring and accurate monitoring, being able to identify what the pest is, what stage of life it's at. Is it going to be a problem? Isn't it going to be a problem? Are there beneficial insects there that can help manage things? And if you do have to step in and do something about it, what and when should you do that? Simple monitoring aids that I use when I'm looking for pests are things like the bug vac, which is useful for pulling up insects and being able to identify small insects that are often hard to see in plants and young plants. Having a hand lens is essential. Most insects, and certainly the early stages of insects, are so small that a hand lens can help you make proper identification. I also use things like pheromone traps. Over on the right side there, the bucket is a Heliothis pheromone trap, a homemade Heliothis pheromone trap. You can buy the pheromones online, and then the bucket is a very simple design just to let you know what's active and when it's active. In the top left-hand corner is a very simple Heliothis egg monitoring system. I just use the hole punch to take out Heliothis eggs, and from those I can work out when the Heliothis are likely to hatch and time any intervention that we need to make to manage them much better. Correct identification in the field can be done with the hand lens and as you gain experience, but also having a microscope for some of the early stages or for being able to identify different species of insect can be very useful. A dissecting microscope with 40 to 60 times magnification is necessary to be able to identify different species of thrips, for example, which I'll talk about a little bit more further on. Being able to identify the particular life cycle and what stage is present on the plant allows you to make a much better accurate plan for when you're going to tackle the pest. Young eggs, a lot of chemicals, say sprays, won't actually kill the eggs of insects, but they are effective against excuse me, against against the early stages of caterpillars or young insects. So being able to time your spray to meet that particular life cycle stage is very important. Sometimes also that includes when will it be a problem to the crop. If it's an egg, it's not actually doing any damage to the crop. So being able, and some adults do much less damage to a crop than the juveniles. So being able to identify those different life stages is very important. The other thing that I'm always looking for as part of an IPM program is any possible biological control. There are many naturally occurring beneficial insects in the crop that can help actually reduce the activity of the pests and sometimes even control them. Being able to identify those insects and which ones are doing the work for you is a really important part of IPM. And then part of that, then the other key part of the monitoring is deciding on what action to take. Are the beneficials managing the pest to a reasonable level? When might things get out of hand, or have they already got out of hand? And then deciding what action you're going to take. Are you going to introduce more beneficials? Are you going to use a chemical? And what chemical might you want to use? So as part of that monitoring and being able to identify different life stages of insects, I've got just some examples of some of key problem pests that we deal with to show you. This is diamondback moth, the key pest probably in most brassicas. The adult is a very small moth that, as you get to recognize them, you'll notice them as you disturb them walking through a crop. But they're very quick and they tend to hide out underneath the leaves. So it's worth being able to know what to look for. The eggs are very small, smaller than a grain of rice. And it takes a while to tune your eye in to be able to identify them. The eggs also change color as they develop. And that's one of the key important parts of the monitoring, is being able to decide whether they're fresh eggs that are going to take 3 to 5 days to hatch, or if they're darker yellow color, they're going to be closer to hatching, 1 to 2 days, depending on temperature. That can help you plan your next action of whether you're going to spray or not. The larvae over on the right-hand side there are quite distinctive. When they're smaller, they have a black head, a dark head, which is often a giveaway at the very earliest stage. These are slightly older instar larvae. And they can be active up from 7 to 10 days, again, depending on the temperature. One of the problems with diamondback and some other key pests is they have high resistance to a lot of chemicals. And their resistance, or their ability to resist some chemicals, increases the larger they get. So being able to target them at a very early stage, soon after the eggs hatch, makes a huge difference to how well you'll manage them in the crop. Diamondback, after the larvae has finished growing, it will develop a pupae that you can see over on the left-hand side there. Usually these are on the plant. They will drop off into the soil to pupate as well, but normally you'll find these on the underside of the leaf in the plant. There is a very good biological control, a parasitic wasp, that you can see on the right-hand side there, diadegma, that's called. They sting the aging larvae, and then as the larvae goes into its pupal stage, the eggs of the wasp start to develop inside it and kill it. And you can tell pupae that have been parasitized by their color. They go this black color, and then you break them open and you'll find a mushy larvae or eggs of the wasp inside. Some other brassica pests and their different stages that you're looking for. Cabbage white, which have been plaguing up this season. Their eggs, again, are small, about the size of a grain of rice. They stand upright on the leaf. Again, they change color as they develop. They start off white and then go towards yellow as they're getting ready to hatch. That can help you make up, decide when you're going to actually tackle them. The eggs take 4 to 5 days to hatch, depending on how warm it is. The larvae is this green, slightly chunky, but also hairy larvae. Cluster caterpillars, again, the eggs are quite distinct, but they get laid in this raft, what we call a raft of many eggs underneath this brown furry substance. They then hatch all around about the same time in again 4 to 5 days time in warm weather. And as they develop, they can make an absolute mess of a plant, as you can see from the picture below. Interestingly, as they grow larger, they become cannibalistic and start eating each other. So you can end up with one large caterpillar where there were many that initially made that damage. Picking the time of spraying is a little bit harder with these, but again, the egg sac or the egg raft do tend to go a darker color just before they're ready to hatch. Something not to get confused with caterpillars in brassicas is vegetable weevil. You can see the larvae in the lower picture there. They are a beetle, not a moth or a butterfly. And it's important that you can identify that as being a beetle larvae, because there are many chemicals that you use for caterpillars and moths that would not be effective against a beetle larvae. They often can be an issue, especially in Wombok for some reason. They seem to like the Asian vegetables. Heliothis is another key pest across a range of crops, sweet corn, cucurbits, even brassicas in years when there are high numbers. Their eggs are quite distinctive again. They tend to lay them in strategic areas on the plant. In the sweet corn, it's usually around where the developing tassel is, and then in the silks or around the close to the silks as the cobs develop. In zucchini, they do lay them on the leaves, but the caterpillar will make its way into the flowers and actually spend some time in the flowers before eating their way out and doing more damage. Their eggs change color again as they develop, taking about 3 to 5 days in warm weather to develop. You can see the 3 in a row there at the top. They start off white, then start to get a darker tinge to them. And then at the end, they have a black dot in the center, which is basically the head of the developing caterpillar inside. This is why I use the hole punch to take samples when I'm monitoring in a sweet corn paddock, especially, so that I can take them back to my office and keep an eye on them and check that my estimate of when they're actually going to develop and emerge is right, and I can let the grower know when's the ideal time to spray for them. Heliothis are particularly difficult to manage as they get larger. The larger caterpillars can shrug off many sprays, unfortunately. Also, getting contact with them in a developing sweet corn crop can be an issue as well. As you can see over on the right-hand side there, given any chance, they'll get into the silks and then drill down into the cobs 2 to 3 weeks into quite a large caterpillar. There is a very good parasitoid of Heliothis, and one way, again, of telling how well that's done is looking at eggs. When the eggs have been parasitized by the small wasp trichogramma, they turn black, like the egg on the left-hand side. And again, this is another reason why I collect eggs from the field, so that I can get an idea of what level of parasitism is going on. Just for another example, over on the right-hand side, that's parasitized cabbage white moth larvae. The wasp that lays its egg on the cabbage white moth lays dozens of eggs, and then as they develop, you can see each of those little packages is a parasitic wasp developing. An example, a good example, another example of applied biological control are the nuclear polyhydroviruses, viruses, MPV viruses. Gemstar and Vivus are a common one of these. They're a virus that is very specific to Heliothis armigera, and they attack the caterpillar, and these are different stages that you can see here of the virus working on the caterpillar. Slowly they kill it. As the caterpillar takes in a mouthful of it, it gets infected, it stops feeding, but then it takes a little while for the virus to work its way through the body of the caterpillar until you end up with one that is completely destroyed by the virus, but also adding to your control by releasing virus into the crop to hopefully get other smaller caterpillars. Being able to identify these also helps, obviously, with the monitoring to let you know how well your control is going. Other pests that it's important to be able to identify accurately would include aphids. There are very many forms of aphids, different colors, but all with a very similar body form. And the main giveaway is the two little spiracles at the rear end, the tailpipes, if you like. On the left-hand side are brassica aphids that have this grey waxy coating on them that can make them sometimes more difficult to manage with chemical sprays. They all have a variety of parasitic wasps that will attack them. And as you can see on those cotton aphids on the right-hand side, the brown extended papery ones are what we call mummies. They're aphids that have been parasitized by a small wasp, and as the wasp develops inside the aphid, it eats it out and just leaves this papery body behind. There are times when parasitism of aphid populations can actually control them, is enough to prevent the aphids from doing damage, economic damage to the crop. But of course, you do need aphids present for the wasps to be present as well. So monitoring and being able to judge the balance of what's enough aphid and what's not too much aphid is an important thing to learn. Thrips are another very common pest and a major issue in certain crops, mainly because they can be a virus vector. Also, when they're in huge numbers, like you can see on that trap or on that cucumber flower, their feeding damage can actually scar the fruit or the crop that they're in and make losses that way. Identification of thrips is important because of this point that they are virus vectors sometimes. Western flower thrips is probably the most notorious vector for tomato spotted wilt virus and impatiens necrotic spot virus, which can be problems in a wide range of crops: lettuce, vine crops, tomatoes, cucumbers, capsicums, chilies, cucurbits of all sorts. The way to actually identify thrips is only really possible with a microscope. Excuse me. For example, on the right-hand side, there is a Western flower thrips. That's at about 60 times magnification. And what you're looking for is the hairs behind the head. If you have hairs on the segment just behind the head there, the pronotum on the front and back of the head, then it's very likely a Western flower thrips. There's a couple of other taxonomic tricks to being able to confirm that, but without that microscope, it's very difficult to confirm thrip species and to know whether you've got a virus vector in the crop or just a plague thrips that will maybe do some feeding damage, but certainly isn't a virus threat. There are, before I move on, I was just thinking that there are useful guides for identifying insects and beneficial insects. AUSVEG has many information sheets AHR has a series of very useful ute guides with pictures to identify pests and beneficials. These are all available from workshops and through local land services and the DPIs. This is a thrips identification guide that New South Wales Agriculture or DPI put out some years ago, which is really very useful. I believe you can still access it on their website. It's also important that you identify beneficial insects and their life cycles and can identify what's beneficial from the different stages. These are just a couple of examples. The brown lacewing on the left there is a great generalist predator in vegetable crops, but its larvae, obviously, which you can see below there, is very different from the adult. But they're both voracious feeders of aphids and moth eggs and other pest insects. Lady beetles also, we're very familiar with the adult lady beetles that we find in the crops, but their larvae look very different from the adult, as you can see in that one above there. But again, you want to be able to identify the larvae because they are just as effective as the adults at feeding on pest insects. As a slight aside, just on the right-hand side, there is one lady beetle that can be a pest, or that is a pest, and that's 28 spotted lady beetle. They all lady beetles lay their eggs in rafts, in groups of dozens of eggs like that. The 28 spotted ladybird is a large orangey colored ladybird with multiple dots on its back, and its larvae are quite distinctive, these yellow orangey spiky looking things. They particularly like cucurbits and can be a problem in zucchini and other field cucurbits, pumpkins, and such like. They are also very fond of thorn apple weed for some reason, where you find them. You can find them a lot as well. They can make a mess of a crop if they're there in largish numbers. So they will need controlling. There are some very useful guides to how to protect the beneficials in a crop by specifically choosing targeted insecticides. These is an example of a group of those useful diagrams from IPM Technologies. You can find them on their website. I think they were produced for, AUSVEG. So they're probably on the AUSVEG site as well. They show what the chemical does to certain key beneficial insects so that you can decide, knowing what's active in your crop, what might be the best control option to use to protect the beneficials and to deal with a growing population of a pest. Another example of this kind of chart, this one I think was put together by Cesar for the grain industry, but same thing, it lists the chemical active ingredients and then a list of the most commonly and most useful beneficials and what the effect of those chemicals are on those beneficials. A few other pests that you need to be aware of and that need particular responses to, so that the monitoring is critical, are the beetles and the bugs. Many beetles, like the red-shouldered beetle on the left-hand side there, aggregate. That means they come in large, they can come in large numbers, descend on a crop, do a lot of damage, and then move on very quickly. Rutherglen bug in the middle. Again, some years Rutherglen bug will plague up and become a major problem. Other years, numbers are quite low and not an issue. The same for the soldier beetle over on the right-hand side there. Interestingly, the soldier beetle, when they're in, are actually predators of other insects. So in small numbers, they can be beneficial and helpful, but they can also plague up. And at that point, their feeding habits tend to mean that they will suck the moisture out of a crop, and they can actually be damaging to the crop in those large numbers. Being able to identify these quickly is really important, because beetle biology means that you need specific chemicals to be able to control them. Normal, just ordinary broad spectrum chemicals won't actually control adult beetles. So being able to identify them and identifying them quickly before they do too much damage is vital to managing them. Another broad pest of many crops are spider mites. There are a few different varieties of spider mite: bean spider mites, tomato spider mites, and the two-spotted spider mite that you can see in the bottom picture there. One of the issues with spider mites is that if you have to deal with a pest by using a broad spectrum insecticide, a very broad spectrum insecticide that kills off any beneficials that are around, you can actually induce a secondary problem with something like spider mites that are very resistant to chemicals moving in and becoming a problem in the crop. This can be true when trying to deal with beetles, where you've had to use a broad spectrum insecticide to take out a descending beetle population. You then need to be monitoring carefully for any signs of secondary infestation from a resistant pest like the spider mite. There are good biological controls of spider mites. The predatory mite over on the right-hand side there is Persimilus. The two orange ones are Persimilus, male and female. And they can be quite effective in the field. We've used them effectively in sweet corn and in eggplant crops right the way through the summer. But you can buy them from commercial insectaries that will sell them. So I think that more or less winds things up. Thank you for attending.